Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have Dr. Louis Falo. He's a professor and chairman. He's at the University of Pittsburgh School of Medicine, and he's working on a new method of vaccine delivery, which appears to be a patch that has a microneedle array, which sounds really innovative and interesting, and we're going to discuss that. And now at a time, of SARS-CoV-2, everyone's looking for a vaccine, a new delivery method may be very interesting for this and other vaccines. So, Louis, thanks for coming.
Thank you for having me.
Yeah. So, tell me a little bit about your background.
Sure. So I am a dermatologist and an immunologist. I did my undergraduate work at the University of Pittsburgh, and then when I went into medical school at Harvard, I developed an interest both in science and in medicine, with a particular interest in immunology. And so during my time there, I decided to spend some dedicated effort developing skills and expertise in immunology, and did a PhD in immunology. And my focus gradually united both my clinical interests and my scientific interests as I became very focused on the immunology of the skin in particular.
Okay. At what point did you conceive of a delivery mechanism using this microneedle array? Was that project your conception, or is it a project you joined?
Yeah, so the idea of, first of all, delivering vaccines to the skin is not a new idea, but it's very rarely used. As you know, most vaccines are delivered with a needle injection into muscle in the upper arm. What most people don't realize, I think, is that the first vaccine, which was one of the most effective vaccines ever, was the smallpox vaccine. And that was actually developed in the late 1700s and was a skin vaccine. So for smallpox, what was actually done was small amounts of the virus was actually placed onto the skin in the form of a droplet, and then they used a needle to actually scratch that virus into the skin, so to get through the outer layer of the skin and to deliver that virus directly into the skin. And that turned out to be extremely effective, not just locally in the skin, but systemically. So it gave a good antibody response that was able to protect people, including their respiratory tract, from contracting the virus. And so because that approach was not particularly reproducible, left a scar, was inconvenient, it gradually faded away, and we went into the intramuscular delivery mode. What I think is very interesting is, the skin is incredibly immunogenic. It's actually, if you think about it, the first line of defense. And so through evolution, it's been designed to be very effective in mounting defenses against invaders. So your skin is constantly exposed to viruses, bacteria, other harmful invaders. And because of that, it's become very efficient at mounting immune responses to these organisms. So the skin can very quickly adapt to the environment, and it contains a really high density of what we call antigen-presenting cells, which are the professional antibody response creators of the immune system. And these cells actually crawl around in the skin as sort of as sentinels or scouts. And what they do is they capture pieces of foreign invaders and then take that into the lymph nodes and program a systemic immune response. And so our idea was to try to figure out a way to deliver antigen effectively into skin that was reproducible, that was safe, and that was actually convenient, so that it could be used for global immunization strategies. And so that's how we came up with the concept of microneedle arrays.
So when you say you're delivering to the skin, I mean, there microneedles that are going through certain layers of the skin. So, just as a refresher, how many layers are there of skin, and which would be a preferential layer to inject, a vaccine, for instance, or is it at multiple layers?
Right. So the skin has three major layers. There's a stratum corneum, which is essentially dead cells that really acts as a physical shield or a physical barrier. Right underneath that is the epidermis, which contains some of these powerful antigen-presenting cells, and then a dermis, which contains both antigen-presenting cells and a host of other cells, mast cells, T cells, other what we call accessory cells that help develop an immune response. And so the microneedle arrays that we developed have microneedles that are obviously very small, micro scale. You can kind of imagine them as being about the width of a human hair, and they're a little more than 0.5 millimeter in length. And so these needles penetrate the outer layer of the stratum corneum of the skin and then enter into the epidermis and the upper layers of the dermis. Now, what's neat about these microneedles is that they're entirely dissolvable. And so the needles themselves are made out of a sugar. And we integrate the target that we want to immunize against, which in this case is a protein from the SARS-CoV-2 virus. And that protein subunit is part of the needle. When the needle is solid, it's very sharp, very hard, and can easily enter the skin. Once it's in the skin, it hydrates very quickly, and the sugars dissolve and release the antigen into these upper layers of the skin, which initiates the immune response. And so it's a very targeted system for delivering antigen very specifically to a microenvironment within the skin.
So is the needle itself the entire needle itself, the body of it is composed of the antigen of the vaccine component?
Yes. So the needle is actually the vaccine. So the entire body of the needle is made out of sugar and the protein antigen. And then there's a backing behind that where you basically think of it as working like a Band-Aid. So you basically apply the patch with some gentle pressure that can be done with your thumb. And that's enough for these needles to actually enter into the skin and then start to absorb moisture and dissolve. So you can imagine this could, in theory, be a vaccine that everyone could apply to themselves. It could be shipped. One of the features of the vaccine is that once this protein is incorporated into these microneedles, it becomes incredibly stable. And so unlike most vaccines, which need to be refrigerated or frozen from when they're made until when they're injected, these microneedle array vaccines are stable enough to be stored and shipped just like Band-Aids. And so for global immunization campaigns, this could be a major benefit.
What's interesting, too, is that the vaccine component literally is fixed in space. So I would think that would make the immune response more effective because the signaling that goes on to attract, our immune cells can go to a localized place over and over and over, and they can build up in this localized place because it's not moving around. So I would think that this is a nice beacon for the immune system, or multiple beacons.
So that's exactly right. And so this is a very localized delivery system where all of the cargo that you're delivering is kept within a very confined space. Part of the advantage of this approach is that unlike needles, where you're literally using force to push a fluid, which causes that fluid to kind of go right through the skin and into the extracellular spaces, this is a more gradual method, and so the deposit doesn't have this hydrostatic pressure and is able to stay in the areas where the needles dissolve. And as you said, that stimulates the local immune response, which then calls in more immune cells and more antigen-presenting cells into that area. So essentially, you've got a little immunization factory going there in a microspace within your skin.
I guess depending on the number of needles, you could probably, I'll call it titrate the immune response. You could start, let's say, with a patch with a few needles. Maybe add a secondary patch at a certain period of time where the immune system gets going, and then you present more of the antigen for it to work on. So it seems like you can again titrate the amount of vaccine presented.
That's exactly right. So you can very finely control the amount of vaccine presented, and it's important that there are much lower levels of vaccine needed with this approach than with traditional approaches where you're injecting into muscle, and that's because of the concentration effect of having all that antigen in one place. You can, as you suggested, you could send out both your initial immunization and your booster dose in the same package in the mail, or you could pick it up at your drugstore and you would have both of those, and the instructions would be to apply one now and then in 4 weeks later apply a second Band-Aid for your second dose of immunogen. I suspect that in the future we'll actually be able to incorporate timed release formulations within the same microneedle array so that we deliver, when we put a patch on, we deliver an initial burst dose and then also a formulation which would stay in the skin and not release for some period of time and then at some point afterwards release so that you could actually get both doses in the same microneedle array.
I guess you could have like a gobstopper effect too, where if the outer layer gets worn away, it reveals an inner layer with a different type of antigen that would, then the body's able to work on that one too. I mean, you could do a lot of things.
Yeah, so that's one of the advantages of technology. And so in addition to putting in an antigen, you could also put in what we call an adjuvant, which is a nonspecific immune stimulant. So you could boost the effectiveness of your vaccine by including an adjuvant with that same antigen. And again, the important point there is that you can use very low levels of adjuvant. So one of the problems with adjuvants, and the reason there aren't in the clinic yet to any large degree, is that they are non-specific immune stimulants. And there's a concern that if you put an adjuvant systemically, you would have off-target effects that might lead to autoimmune effects or fevers that would obviously be undesirable, basically non-specific immunity. That risk is greatly reduced by using extremely small doses and keeping them localized to the skin. So combination agents are definitely a possibility, and you could even imagine getting multiple immunizations within the same patch. So your next flu vaccine could also be a vaccine against coronavirus at the same time.
The needles themselves, the microneedles, do they get passivated by the immune function of the body and covered up, or how do they work to make sure that they have a useful life?
Yeah. So the needles themselves are completely inert. So they're made out of a very simple sugar, and they actually completely dissolve. And that sugar, once it's solubilized, just ends up in the interstitial fluid and gets carried away. So the substrate of the needle itself doesn't have any activity. The activity is in whatever cargo you choose to deliver. And these needles are already in clinical use, and so our group is involved in actually 2 clinical trials. Both of them are actually focused on skin cancer. And so in those applications, for example, we actually deliver a chemotherapeutic agent directly to skin cancers in patients, and we localize that chemotherapy to that specific skin cancer, and then we deliver it in such low doses that it doesn't have all the adverse effects that you see with systemic chemotherapies that are used for several other types of cancer. So that gives us hope not just for the vaccine application, but also for the cancer application. And I think that so far we've shown that this microneedle technology in general is safe and is effective in delivering cargoes in patients.
Is anyone contemplating using a microneedle patch in conjunction with surgery? Let's say you have to work on a given organ, the liver. Could you open the person up and put in a patch itself onto the surface of the liver? So it would act locally, but you couldn't normally get to it through the skin.
Right. Yeah, so exactly that concept is something that's under development, and so whether that's a patch or the same substances of a patch that is deposited by a needle that's able to get into the liver without actually opening a patient up, the same concept of having something which is in a substrate that is solid and able to penetrate so that it reaches the target and then dissolves and releases its cargo is something that's definitely of interest to several groups right now as a new way to deliver drugs to very specifically to different organs within the body.
Yeah, this is really interesting. Are there any patches that are being developed that they'll act as a scaffolding for for tissue to grow, or they just have a specific geometry that was the geometry of this microneedle array, did it have to be played with a lot? Did you have to have certain spacing and depth and number, et cetera, to make it effective?
Yeah, so if you can, so that's actually a terrific point. And so when we started down this path, we were sure to optimize both the needle geometries and the geometries of how the needles are arranged on the patch for penetration into human skin. So if you can imagine, the bed of nails effect works against you. So if you have your needles spaced too closely, if they're not sharp enough, you get there's a lot of elasticity in the skin, and you just end up pushing the skin down rather than puncturing the skin. And so you have forces working against you there in that the thinner and sharper that a needle is, the less volume it has that you can deliver. On the other hand, as the needles become thicker and of different geometries, they're more difficult. It's more difficult for them to penetrate into skin. And so we basically now are using two types of geometries. One is an obelisk shape, so think the Washington Monument, which has a very sharp tip and yet has a tapered body so that it's able to hold considerable amount of cargo. The second geometry that we really enjoy now and have high hopes for is one that's actually been enabled by 3D printing. And it basically is like an arrowhead. And so if you can imagine an arrow where the tip of the arrow is very sharp and contains your cargo, and the shaft of the arrow is made to dissolve very quickly, you can basically, once you apply pressure, the arrowheads will penetrate into the skin, the shafts will quickly dissolve, and the arrowheads will remain in place in the skin where they can be a depot for drug release. The same type of technology has been used and is under development for wound healing, for example, so that you can create a scaffolding for reepithelialization of wounds. And I can envision several other uses that could be developed in the future.
Yeah, this is really cool. Do you own all the patents on this, or are you just working on it?
Yeah. No, we sure do. So there's a very robust patent portfolio that is protecting microneedle design, microneedle delivery, and then each of the various use applications. We have spun out one company for the cancer application. The name of the company is SkinJect, and they are now in clinical trials. And we are rapidly progressing with the SARS CoV-2 vaccine that we are developing now, and hope to have that in the clinic in the not too distant future.
When the patch is on someone, how long do they keep it on? And then when they remove it, do they just pull it off and it really comes off easily like a Band-Aid, or what happens to it?
Yeah, so think Velcro. So I've been told it feels a little like Velcro. And when you take the patch off, first of all, the patch is only on for minutes. So 5 minutes, 10 minutes to be super sure that you've delivered everything, but realistically, probably 1 or 2 minutes is sufficient. When you take the patch off, the drug is no longer there. So all of the drug is loaded in the distal ends of the needles, so the part that goes into the skin first, and that has dissolved and is in the skin. And so all you're left with is basically sugar on a Band-Aid. So there's no harmful waste here. You can just throw that in the trash like any other Band-Aid. And the other interesting thing is, because the needles are so short, they don't cause any pain, and there's no bleeding because they aren't long enough to reach blood vessels or nerves. And so it's a pain-free immunization approach.
Yeah, I've worn like a glucose, continuous glucose monitor. They have a microneedle, which yours probably are thinner even, I don't know. But that one doesn't really hurt either. It's maybe like itchy for a second, but there's really no pain.
Exactly. Just some pressure is usually what happens. Now, the one side effect that we might see, and we haven't done vaccines in patients yet, but the one side effect that we might see is if we're very successful and we induce a robust immune response, we might expect to see some erythema at the site over the next day or two. But to us, that would be a very positive sign in that the immune response in the skin was actually working. And I doubt that it would be any more uncomfortable than maybe at worst a mosquito bite.
Of the needle array, are there a few of the needles that go deeper to trigger more of an immune response, or is it good enough that they all go to into the epidermis, or do you have to drive any deeper?
So the needles that we're using go through the epidermis and into the upper dermis, and that's the ideal place to deposit the antigen, and so they're all the same size. The technology is incredibly flexible. We can make them as long as we need. So if there were other applications where we wanted to go deeper, potentially even into the fat layer underneath the skin, that could be done. We haven't done that yet, and I think in those use cases you probably would see some pain and bleeding because the needles are longer at that point and they're able to reach some of those other structures.
So the skin of anywhere in the body, does it? I guess there's probably multiple factors that make it a great place to cause immunization. I guess the body's used to providing an immune response to the skin. It's a huge surface area. I guess the full ability of the immune system probably would be best used there because that's the spot where most of the battles happen, I guess, on a continual basis.
Exactly. Every day you're under attack. So viruses, bacteria, other harmful invaders are trying to get to you, and your skin is protecting you. And so it has this first line of defense, which is just this barrier stratum corneum that we talked about a little earlier. And then after that, it has an epidermis, which is layers and literally looks like a brick wall of cells that we call keratinocytes, which are also very important for barrier function, and antigen-presenting cells mixed in with them. And then deeper than that are the other cell types, including different types of antigen-presenting cells that are more specialized, as well as accessory cells like mast cells, the cells that are involved with giving you hives, for example, and other immune stimulators. And so all of these systems are very well-controlled networks that work together to generate an immune response. And one of the remarkable things is that the immune response that is enabled through the skin isn't just a skin immune response. It's actually a systemic immune response because these antigen-presenting cells actually are conditioned based on what's happening in the skin and then circulate into draining lymph nodes more centrally where they make the responses of both T cells and B cells and antibodies, which then distribute throughout the body. And so, it's fascinating that you can immunize in the skin and then find specific antibodies and T cells in the lung tissue against the antigen that you immunized in the skin. And that's just part of what evolution has developed to protect us.
Are there certain parts of the body that are more amenable? Like, is the arm a good spot, or the thigh better, or back of the hand?
all of those spots would work. I think the biggest issue is you want to look for a non-hair-bearing spot, or not one that doesn't have a particular large amount of hair, because the hair would actually create a physical barrier to the needles to some extent. We're intending this to be upper arm for a vaccine, pretty much in the same spot that you would get an IM needle injection. However, we've already learned from our skin cancer experience that this can be anywhere. Skin cancers can appear anywhere, and thus far we've treated multiple sites, face, back, legs, etc., and the microneedles appear to be equally effective in all those sites.
So clinically, is this in use, and when will people, you think, see it be used extensively clinically?
Yeah, so clinically right now it's in clinical trials, so phase 1 clinical trials for cancer. After phase 1 trials, as many folks have heard, the testing process then involves phase 2, which is a much larger number of patients, and then phase 3, which is a very large number of patients, and all of these are assessed for safety. As far as vaccine applications, there have been 1 or 2 phase 1 clinical studies with different types of microneedle arrays. So it's starting, the technology is starting to get into the clinic. We would hope that our vaccine will be into the clinic before the end of the summer. And once we get started, we've designed the trial to advance very quickly. So I would hope that we would be able to progress through those phases at a fairly rapid rate, assuming that everything goes as expected and we don't see any adverse events or safety issues that would need to send us back to the drawing board.
Is a microneedle array patch used clinically for anything right now, or is this a race to get into?
It has not yet been clinically approved for any applications at this point.
I guess it's funny, if the first one you do works, that'll open the door for faster adoption of any microneedle array technology, or the technology used for other types of interventions. But
yeah,
it's weird. Do you pick the one that you think is most likely to work so it can open the door and make the other ones faster, or do you pick the one most needed?
Yeah, so it's a combination of the two. So I think the skin cancer application is an application that has a very high likelihood of success that would open the door for this technology. And then obviously the vaccine application efforts accelerated dramatically when COVID hit us. So we had already been working on microneedle array delivery of other vaccines. So we had already published papers with microneedles for Zika virus vaccine and one of the SARS-CoV-2 relatives, the MERS vaccine. And that's one of the reasons that we were able to so quickly get the microneedle needle array technology into the animal models for SARS-CoV-2, is that we had these active vaccine projects already underway.
Well, very good. What do you think is the timeline? What's the window of, how long till this could actually be used clinically?
Yeah, so from the vaccine standpoint, we would hope to begin clinical trials, a phase 1, maybe phase 1/2 clinical trial by the end of the summer. And then the progress would really depend on how well the vaccine works. And so safety is always the first and most important concern. And assuming the vaccine is safe, we'll be able to accelerate quickly through the phase 1, 2, and 3 studies. If the vaccine is effective, obviously, that would be great motivation, not just for moving quickly through the trials, but for scaling the vaccine to being able to manufacture the multiple millions, if not a billion, mRNAs that would be needed for global applications of an mRNA vaccine for COVID-19.
Mm-hmm. Well, very good. Louis, what's the best way for people to find out more and keep tabs on your progress?
Yeah, so you can check our departmental website at the Department of Dermatology in the University of Pittsburgh. We update it regularly with progress on this project, microneedle arrays in general, and the other work that's going on at the University of Pittsburgh.
Well, very good. Louis, thanks for coming. This is really, really cool. I found you because of an article that came out, and I'm glad that you were able to take the time to talk. So this is great, and I hope it works.
Yeah, thank you very much for inviting me.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.